Okay, so this is simple columnar epithelium. You can see the column-shaped cells. The nuclei are either round or oval. In this picture, they're more oval. And then you've got roundish, opaque cells. Those are called goblet cells. If we look at another example of simple columnar, again, column-shaped cells, goblet cells as well, found within that tissue. A third example, again, same type of thing. And again, if we're pointing to any of the slides and it's pointing to the nucleus, you can say nucleus for that structure. Okay? So it's the nucleus of a simple columnar cell. And let's go here. Now, when we look at pseudostratified, pseudo means fake or false. And so it looks, pseudo means fake, and stratified means multiple layers. So it looks like there's multiple layers because the nuclei are at different levels. So they're kind of all over the place there. And so that's pseudo-stratified columnar. Don't forget to say columnar because they are column-shaped cells. And all of these weird kind of shaped opaque cells, those are goblet cells inside there. And then on top you have the structure called cilia, which are those hair-like structures. Look at another example. Same type of thing. Got the hot mess of nuclei all over the place. You've got the goblet cells and the cilia on top. That is pseudo-stratified columnar. And there's a third example of pseudo-stratified columnar. All right, now this one is stratified squamous. We know that simple means one layer, stratified means more than one layer. So we've got, and squamous means flat cells. So stratified squamous is multiple layers of flat cells. We're looking at this top layer here as our good example of the stratified squamous. And stratified squamous can be keratinized or non-keratinized. Now, you won't have this here, which is esophagus, but if it's in a mucous membrane, then it's going to be non-keratinized. If it's in thin or thick skin, it's going to be keratinized. And so we look for like big flaking on the top, which we don't see, which we would see in thick skin. Thin skin, it gets very not so dense in here, right? Very much less dense. So if we're dense and we don't have any big flaking, then we generally look at it and say it's non-keratinized. Okay? We'll go to another one here. Now, you may look at this and go, well, that's kind of spacious. It's not. These are the cells. They're still packed together. So still densely packed. There's no giant flaking coming off the top here. So that is non-keratinized stratified squamous. If we look on that, this is actually the exact same slide, but a closer up view, so you can kind of visualize the actual cells. And this is one more view of stratified squamous. All of those were non-keratinized. Now this is our first example of a keratinized stratified squamous. See those flakes, those big flakes coming off? So that's very clear that it's flaking. It's thick skin. You'll see the difference when we look at thin skin. And so on thick skin, you're going to see the flaking. It's also going to be very dense. Okay? When you look here, another example of thick skin. And so you've got that big chunk coming off the top there. And again, this is thick skin. You can compare to the thin. The tissue, though, that we're looking at right here, stratified squamous epithelium. And then this is thin skin. And you need to know the difference between thick and thin, but again, we can see that there's a big difference between those two. Now this, I don't really see the flaking very well on this, but look how non-dense that is, right? So you won't really see the cells packed together on that. And then the same type of thing here for thin skin. Remember, all skin is keratinized, so not very dense in there. Regardless of flaking, doesn't matter. Okay. And so let's go for that. Let's go to simple cuboidal. Cuboid cells usually make a lumen, and we can see an oval lumen here. It's actually a string of cube-shaped cells, right, with round nuclei that make a lumen. Remember, the space inside a tube, or the cavity in a tube, is called a lumen. So if I point it here, we would say lumen. And if I point to the cell, simple cuboidal cell, but tissue, simple cuboidal epithelium. Here's another example. Now, the thing on this, how do we know these are cube-shaped cells? Well, they're not columns, they're not flat like squamous cells, they're not dome-shaped like transitional, and they have nice round nuclei. And again, they're making a lumen. So these are all indicators that this is a simple cuboidal epithelium. Another example is here. This, you can see some of the cell walls on this a little bit better. And so again, simple cuboidal. If we want stratified cuboidal, remember stratified means more than one cell layer, we look at the nuclei stacked on top of each other. Still making a lumen here, but the nuclei, we see the multiple layers. So that means there's more than one cube-shaped layer. They're definitely not columns, and they're not flat either. So we know that must be stratified cuboidal. We have some other tissue out here we're going to go over at the end of this chapter. Okay, these are transitional cells. You see how they're dome-shaped? They're rounded off. Look for the rounded off shapes. That'll help you identify transitional epithelium, which is the extensible tissue, right? It stretches, and so that's going to help us with that. It kind of falls in its own class, so it's not stratified or squamous. Here's another example of that. This isn't my favorite example because you can't see the rounded edges as well on this, but this is another example of transitional. Then you have this one here, which you can kind of see again, those dome-shaped cells that we're looking for. As it starts to stretch, they become more flattened out, but we look for the dome shape throughout. Simple squamous, if we look at the inside layer of the artery, it's the single cell layer that lines the artery right through here, okay? And so it's just that one cell layer. This is another example of simple squamous, but now we're looking at it like an S to I view. We're looking down, this is like a sheet of single cells, one single cell sheet. And so you've got nuclei that you see throughout, right? So some of them on the insides. That's going to be an indicator to help us differentiate from adipose tissue. It's homogeneous. You don't see any spaces in between there. It's like a cobblestone street, right? All strung together, a nice sheet of cells. Has to be simple squamous. By the way, they're very irregular. This is not a cube shape. It's not a column. It's not a dome shape. Again, it falls into our category where we have to say it's got to be a simple squamous, just looking at it from an S to I view. If we go here, we're looking again from that side. We're looking from the side. We've got the single cell layer. So this is a venial, which is a small vein. The inside layer is what we're looking at. Disc-shaped nucleus, and that's going to be an indicator as well when we're looking from the side. If we go back to this one, they're disc-shaped. You just can't see the side. You can't see how they're disc-shaped, right? So that's the thing you have to deal